In this section, we're going to talk about the detailed steps that are involved in the development of most vaccines. As I'll emphasize several times, not every vaccine is developed in this specific timeline or formula, and some of the steps are skipped over or done out of turn based upon the data that are available from the scientific studies that have been done to date. Earlier we talked about development of the first inactivated polio vaccine. If we go back in time to the 1800s, it's when epidemic polio was recognized as a disease and a disease that warranted possible intervention. In 1908, there was an initial animal model developed here at the School of Public Health, which helped understand how the virus was transmitted and some of the risk factors for development of paralysis. In 1936, the very first polio vaccine was developed. Unfortunately, the vaccine was incompletely inactivated, and wild-type virus was actually administered instead of an inactivated vaccine, and many children developed polio as part of the clinical trials that were involved with that vaccine. It wasn't until 1949 that an effective means of preventing polio was identified through the use of passive immunoglobulin, or IgG prophylaxis as it's listed here. Also in that year, John Enders and his team was successful in growing the polio virus in tissue culture, which was a landmark development that eventually led to the development of a vaccine, and John Enders and his team received the Nobel Prize for growing the virus in tissue culture. It was only 5 years until the first vaccine was developed by Jonas Salk, and 1 year after the vaccine trial was begun, the results showed it was highly effective and put immediately into general use. Nowadays, there's a much longer delay from the completion of a field trial to the widespread use of a vaccine. In reality, there are many thousands of individual steps and decisions that are made during the course of developing a new vaccine. I've simplified this process in the next few slides to show you the major steps. For many of the vaccines, innovative investigators were able to skip over some of the steps, and some vaccines like hepatitis B wouldn't have been developed if they hadn't done that. The first step, which you heard about earlier, is recognizing the disease as a distinct entity. Smallpox had to be differentiated from some of the other rash illnesses, such as measles and chickenpox, and I can tell you there was confusion between these illnesses. Even during the smallpox eradication program, there were problems sometimes in distinguishing chickenpox from smallpox. Once you have the distinct clinical disease, you ideally need to have, or would like to have, the etiologic agent identified by growing it in the laboratory, and then establish an animal model for the disease so that you've got a way to test whether or not a candidate vaccine might protect against that infection or the disease associated with the infection. We have different animal models for different diseases. For instance, for influenza, probably the best animal model is the ferret. For measles, rhesus monkeys were used. We don't have an abundant supply of rhesus monkeys these days, and so people are trying to work with much smaller animals for the most part. For HIV, the best animal model is a chimpanzee, so they do clinical trials with 1, 2, and 3 chimpanzees, far different than doing studies in small mammals. For some diseases, such as the meningococcus, there was no acceptable animal model, and the entire development was done using only humans for all of the testing. For hepatitis B, Saul Krugman and others skipped the isolation process and the identification of the agent. They didn't even grow the virus in the laboratory before they had a successful vaccine. They proved that human plasma was the source of the infection, and they were able to inactivate the plasma, and the initial vaccines were derived from human plasma well before they could grow this agent and grow it in any kind of a tissue culture through the traditional methods. When we're dealing with individual antigens, instead of the whole organism, mice are often used for studying the immune response because the mouse immune system has many similarities to the human immune system. However, We've been misled in a number of cases, and a famous quote is that mice lie, and that comes from the fact that there have been many vaccine candidates that seemed to work and looked like they were going to be very effective based upon mice studies, but then they just don't work in humans. So there's a lot of discouragement, and unfortunately most of the immunologic research that is done in mice for practical reasons. Moving on, we need to then inactivate or attenuate the agent in the laboratory, or identify the individual antigens that induce protective antibodies. There's a new modern process called reverse genomics, where they actually are searching the entire genome of organisms to try to figure out which proteins can then be used to induce a protective immune response. Big shotgun approach, where they sometimes test 1,000 different proteins and then come up with a half a dozen candidates and subsequently narrow it down to one or two. You then need to develop a candidate vaccine, and there are strict procedures developed by regulatory agencies in the United States. They're called Good Manufacturing Practice by the FDA. You can't just take a product that you've developed in my laboratory, anybody else's laboratory, and just start putting it into humans. You've got to have very careful documentation at every step of the process that you have avoided getting contamination or something in there that shouldn't be there that might be harmful. Then, if you have an animal model, you evaluate the candidate vaccine for its ability to protect that animal against infection or disease, and ideally identify a measure or an immunologic correlate of immunity against the disease, so you know how much of an immune response you need in order to protect that animal. We often don't have that for humans at the time when the clinical trials are begun. You then have to go through the process of developing the detailed protocols for studying humans. You have to have approval from the Food and Drug Administration and get what's called an investigational new drug, or IND, in order to conduct those clinical studies. First studies are phase 1, where we study safety and immunogenicity, and often then try out different doses to find out just how much you need. Moving on to larger scale studies with phase 2, we do safety and immunogenicity. Then in phase 3 trials, much larger for efficacy and safety. And sometimes there's an embedded portion of these studies where they also draw some bloods to try to get a measure of how much of an immune response is correlated with protection, but that's not essential. Applying to the Food and Drug Administration has become a relatively cumbersome process with a small room full of paper. It used to be, and now they've developed electronic mechanisms for this, but it's a huge process to prepare all the materials that need to go through and all the documentation of every step that you have followed in the development of these vaccines. This is just a copy of the FDA website documenting the steps that you have to go through to apply. The FDA takes up to a year, they have, for the initial review, but if they have questions and it goes back, and they sometimes insist upon additional studies, they say you're not ready yet, you have to do this or that, another thing, and it sometimes has taken 10 or more years, like for varicella vaccine, to be developed. If the FDA approves the vaccine and grants a licensure, then advisory committees review the vaccine and make recommendations. In reality, they actually start when they are given a signal that the vaccine is likely to be approved by the FDA, so it's oftentimes a year ahead of time that the advisory committees start making recommendations or developing recommendations. They don't finalize them until the product is licensed. After licensure, the company goes through a marketing process, and also in recent years, the FDA has insisted upon setting up what's now called phase 4 studies for surveillance to monitor the safety after vaccination and to document that the vaccine is still effective or potent after licensure. This is a very long and complicated process. The usual process for a vaccine takes between 10 and 15 years. Many candidates fail for every one that's successfully developed. Most of the failures are identified in the preclinical or early clinical phase 1, phase 2 studies, By far the most expensive of the studies are the phase 3 trials, which cost tens of millions, sometimes $100 million or more. And so if a product isn't going to work, the manufacturer wants it out of their development chain as early as possible in the development process. Most recent estimates of the total cost to develop a new vaccine are somewhere between $100 million and $1 billion for each successful vaccine. Now that incorporates the failures as well as the successes. Manufacturers don't release this information. It's considered proprietary or confidential. A lot of the early cost in identifying antigens or potential candidate vaccines are incurred by NIH or other sources of funding at academic centers, such as here at Hopkins and elsewhere. There are a few exceptions, and one most noted is the recent development of the conjugate meningococcal A vaccine for use in Africa, which was developed almost entirely with funding from the Global Alliance Vaccine Initiative and the Gates Foundation. And it's remarkable that they're developing a vaccine that will be sold for only 40 cents a dose because they've basically used public funding at all steps, and it's almost a not-for-profit process in terms of making the vaccine. There has to be a small amount of profit for the manufacturer, but that's a remarkable achievement, at a cost of 1/10 to 1/100th what the costs are for most of the other recently developed vaccines. Now in the United States, we do have 15 different diseases that we vaccinate all children against, so we've been highly successful at developing vaccines. There's some noted differences in these vaccines, such as hepatitis B. We start at birth. We give 3 doses, completed ideally by 6 months, but certainly by no later than 18 months. Other vaccines, such as measles, mumps, and rubella, which are combined, first dose isn't given till after 12 months. And there are important reasons for this. There's interference by maternal antibody with these live attenuated vaccines. We've recently added hepatitis A for all children. That's a 2-dose schedule. The variability in the timing of these vaccines, the doses, the number of doses, all was developed based upon the clinical trials that the manufacturers conducted. And they oftentimes tried other things, including starting earlier, but it didn't work. And so for vaccines like hepatitis A, we have to start after 12 months of age. As we move to older age groups, this is the current United States immunization schedule for 7 to 18-year-old children, where we give different vaccines. We give a single dose of TDAP, which is different than the pediatric DTAP preparation, smaller amounts of several of the antigens. We also give human papillomavirus vaccine, 3 doses. Initially it was just for girls, and now it's for both girls and boys. So everyone should be immunized against human papillomavirus. We give a dose of meningococcal conjugate vaccine routinely now at 11 to 12 years of age, but sometimes some children aren't getting it until 13 to 18 years of age. If they get the dose at 11 to 12 years of age, they're getting a second dose 16 to 18 years of age, especially just before they go to college when there is some increased risk. Everyone should be getting a dose of influenza vaccine. And then we have catch-up and special risk groups as shown in the other bars on this slide. For adults, the schedule continues to call for an individual dose of influenza vaccine for everyone. And it's catch-up for other vaccines. They get a dose of Tdap if they haven't had one before, or it's TD if they've already had a single dose of the Tdap. And catch-up for varicella vaccine. We have a recommendation for a single dose of pneumococcal polysaccharide vaccine. There is a recently licensed 13-valent pneumococcal conjugate vaccine, which studies have shown to be effective in adults. And there's consideration for allowing for the pneumococcal conjugate vaccine instead of the plain polysaccharide vaccine, but sometime in the near future, the advisory committees will decide on that, and there may be the option of giving either. In the World Health Organization expanded program on immunization schedule, there is some variability with regard to the possibility of when you introduce hepatitis B vaccine. But note they also give a vaccine against tuberculosis, the BCG vaccine, at birth, which we do not do in the United States. Also, in many countries, especially in Africa, they're starting earlier with measles vaccine at 9 months of age. But as measles has been eliminated in the Western Hemisphere, it's now at 12 months of age. And hopefully, as measles gets eliminated from many other areas of the world, it'll be back to 12 months of age. But there are compromises with regard to when one starts the vaccination. They start earlier in Africa because the disease occurs at an earlier age. And now WHO is also giving a second dose of measles vaccine, but I won't put that in here. That's part of usually campaigns that are done. There will be new vaccines added to the WHO schedule in a recommendation that rotavirus vaccine be introduced. There are 2 different vaccines out there. They've not yet been introduced in most countries in the developing world. And through extraordinary efforts that are ongoing through Gavi and program here at our School of Public Health called PNEUMO-ADIP, efforts are being made to introduce pneumococcal conjugate vaccines in all developing countries. They're already underway in several of them, and there are major hurdles to be overcome with introduction of these vaccines. Many of you in your public health career may be involved in studies or programs to help bring these newer vaccines, which are quite expensive, into widespread use in developing countries, and do some of the additional evaluations that are going to be necessary to identify the optimal timing or effectiveness of the vaccines and overcoming some of the issues like decreased effectiveness of rotavirus vaccine in developing countries. I wish you luck. I encourage you to pursue your interest if you have them in vaccines. We have several other courses on vaccines that are taught, and you can complete a certificate in the field of vaccines. I teach a course in the second quarter called Vaccine Development and Application. If you are more interested in the development process, and we'll get into detail in many of the different vaccines in terms of how they were developed and how they're used. Thank you very much, and I wish you the best in your academic career.